everyone, I'm Jenna.
And I'm Mark. Thank you guys so much for joining us for yet another episode of Cincinnati Zoo Tales. Jenna, we're being joined by another guest, I should say, who is much smarter than either of us.
We always say that when we
I'm smart in a different way.
They're always so nice about it.
It's always fun for us, though, because we learn a lot from these episodes. It's always exciting. We're being joined by Louisa Rispoli, who is one of our postdoctoral scientists at CREW over at the Center for Research of Endangered Wildlife. Thank you so much for taking the time out of your day. We know you're busy.
I really appreciate you inviting me. I'm really excited to talk to you about all the things I do, and especially things about hippos.
Yes, it's funny because we've talked about this before, but we don't really understand what a scientist does on a daily basis, and all of you have different roles.
Right.
And, I don't know, it's so fascinating to me that you guys grew up and somehow ended up knowing this was a job, because I wouldn't have even known or come across that.
Yeah, I mean, my path it's a really interesting one, because I hadn't planned to go into research.
Okay.
I mean, like you said, I didn't even really knew it existed. I was going to go to vet school, and then I needed a job while I was an undergraduate. And I got somehow into a fruit fly lab.
Oh, okay.
Taking apart fruit flies, looking at their genetics and things like that.
Oh my gosh, taking apart. They're already so tiny. How do you do that?
Very under a microscope with tweezers. But yeah, so I started reproduction very early because I was working with fruit fly ovaries.
Wow.
So this was undergrad?
This is my undergrad.
Wow.
Where did you go for undergrad?
UC Davis.
Okay, wow.
So I'm from California. And that's where I learned, I like being in the lab. I like asking questions that we don't know the answers to. And, I mean, I didn't really like fruit flies. So I knew I wanted to go slightly bigger.
Yes.
And so I went and did my Ph.D. at Colorado State.
Okay.
And I was really focused on, it was still reproduction, but more kind of like the hormones, we call it endocrinology, and how the brain talks to the rest of the part of your body to kind of control fertility and things like that. So, and I was trained more really thinking about human reproduction, but a lot of domestic animals is what we use for models, so we can understand how to develop like contraceptives or how to sustain a pregnancy and things like that.
Which is funny, that came in handy later on. We'll talk about that.
So I was kind of cross-trained to think about reproduction as a whole, and learn that there's going to be individual differences between species, but think about the things, the little black boxes that we still don't know the answers to.
See, and that's what, I just don't understand how you can find out the answers doing the things you do. But were you initially, growing up or in undergrad, wanting to work with animals?
Oh, yes.
Okay.
Oh, yes. Like I said, I wanted to go to the vet school.
Okay.
I wanted to be a mixed practice veterinarian.
Okay.
I wanted to do both large and small and everything in between.
Okay.
And I volunteered. I was really excited. I volunteered at a vet hospital while I was at UC Davis, who specialized in reptiles. And I thought that was really
Very
cool.
Yeah.
So I didn't have a lot of hands-on. I did help my friend with his 25-foot ball python.
Wow.
But I just found it fascinating, and I love the idea of taking care of animals. I do it from the science side to try to develop tools to help them sustain themselves, help them to thrive, help their health. And I focus on reproduction. I focus on fertility. I focus on both sides of the coins. Sometimes we want them to be very fertile, and sometimes we don't. And trying to understand when we can't get it to work the way we want.
Yeah. I mean, so you were at Colorado State, and where did you go from there?
So I went to Tennessee. I was at University of Tennessee, and that's where my first postdoc.
Okay.
And I was working mostly cattle fertility. And this is where sperm comes into it, the male gamete. And they needed somebody who had a really good grounding reproductive physiology, but no preconceptions about sperm. And that's what I had, because I was focused on hormones, not sperm.
Okay.
But when you get trained. I know general physiology. I know how, why you need to eat in order to sustain a pregnancy kind of thing. You need all the parts work together. It's kind of an ecosystem just in one body kind of thing. So I understood about sperm, so that's what I did for 15 years, was studying cattle sperm. So I know a lot about sperm.
Oh, 15 years, what are you studying? Are you, what are you looking for? What are you trying to learn?
One part of our lab was looking at an in vitro production and trying to understand, for dairy cows, they get hot and they don't get pregnant. Well, we kind of want milk all the time. So in the summer, it's not really good if we don't get cows pregnant because you have to have a pregnant cow in order to get milk.
Oh.
Sure.
So part of the thing is, making sure I handle sperm to fertilize an oocyte and, and understand that connection and why heat stress may compromise the oocyte, that female gamete, and not produce an embryo. So I got a lot of work with that. I also just studying the proteins of sperm, what controls its fertility. I'm a molecular biologist, which means I tend to take everything apart and make it glow. And so I've done that a lot of sperm. So that's why, I started working, I had by chance to work with Memphis Zoo and work with their elephants and trying to understand why they weren't getting their females pregnant. And I was interested on the sperm side that maybe, something was going wrong when we did these artificial inseminations. And I'm like, I kind of want to do this all the time. So I took a step back and I'm changing my career path, and that's why I'm here at Cincinnati Zoo.
Which that's brave, I think.
Incredible,
yeah, and just you've done so much for so long, and then just switching is very neat.
I'm still doing all the same techniques, all the same kind of research. My species has changed focus, and I'm much happier. I'm much happier. I love what I do.
Good.
So how did you get involved at Memphis?
So just kind of a connection between my mentor there at University of Tennessee.
Okay.
She had gone to school with somebody who was at Memphis Zoo, who was the director at the time. And so he had come to talk to our classes for our advanced repro class, and then invited a group of us to come and watch an artificial insemination in an elephant. And so just because he was letting us look at the sperm, I'm like, hey, have you thought about this? And so it developed into a small project, and I decided I wanted it to be, instead of 2% of my time,
all of it,
100% of my time.
Awesome. Did you end up figuring anything out for their elephants or making any sort of big
So what I was looking at is whether when we were doing the artificial inseminations, if they were causing an inflammation reaction in the females, and so that subsequent artificial inseminations, maybe they were having an immune reaction and that was preventing them from getting pregnant. And in the 1 out of the 4 of the females, there definitely seemed to be something going on. It was a really small study, and and whether that is the problem, we still haven't really figured out 100% how we, do artificial insemination in elephants and produce a pregnancy all the time. But it's a little bit of data, and we're working on it. I'm actually working with Denver Zoo to try to understand more about Asian elephant semen and how we cryopreserve it, how we freeze it. And so hopefully that will help us, if we freeze it better and make it easier to do artificial insemination and stuff.
So do you freeze it a certain way for a certain amount of time, and then you thaw it and you see how healthy it is, and then you freeze it a different way and then see if it's healthier that way?
It's exactly what we do.
Oh my gosh. But then you just have to come up with the different ways.
Yeah. We have to, it's amazing because we have a wealth of information from horses and from cattle and from humans. And human sperm is not as healthy as some others. So we've actually learned a lot of lessons. So we're taking, instead of animals modeling for human health is now modeling for animals.
That.
Interesting, yeah.
I've never thought about it needing to go back that way.
Yeah.
And it is interesting because a lot of the species we take care of in zoos, they don't have a whole lot of data. There's very little out there, so every bit you can grab matters, right?
Which I think is interesting because some people, most people have rolled with it. We joke that Bibi's birth control is on the Fritz. And we have baby Fritz here with us. But when you think about it, humans, we can study them so much easier and know so much more about them and all these different types of birth control and all sorts of things. And people still get pregnant while on birth control.
Yeah. So a lot of times that somebody forgets that something is going on kind of thing.
That's very, very true. It's usually human error. But
it's a really good point because, with humans, we don't have one size fits all.
Yeah.
If you look at the market, there's 16 to 20 different forms of birth control. And plus there's different levels within that because we know we have averages. Mm-hmm. So we take a group and we sum down the numbers and we say, on average, this will work. Mm. But we know that average isn't going to work for the somebody on the low side or somebody on the high side. And I have a feeling Bibi maybe is a unique individual who is not going to fit the average.
Yeah. Right. And also when you think about it, there are so many humans, and there's only about 90 hippos living in zoos in North America in general. And then you half that by male, female. And and then the ones that are able to participate in any sort of study, which I don't even know if there are, birth control studies, there's just like, here, this has worked for cows or horses
Right.
or, whales or whatever, and then we're going to try and transfer that over to hippos.
Right.
So there's still so much to learn.
And I mean, we are trying. We do have the reproductive contraceptive center who's collecting information. So all the zoos that are participating in like giving contraceptive to different animals, they are trying to have guidelines and say, hey, this seems to be effective and this seems to work. But it is still kind of, we're not really doing research. We're kind of trying out based on, okay, so we know this species is close to horses, so let's try what works in horses. We know this species is close to cattle. Let's try what works in cattle. I mean, with primates, it's kind of easy. Okay, this species like human.
Yes, it makes it easier.
But it's not perfect.
Right.
But at least we always say, okay, you make sure you take your dose every day.
Yes.
Which humans, we're still working on that.
Yeah, the animals might actually have more accuracy.
Yes,
we do.
We get in trouble at our job if we don't. But so you ended up at Cincinnati, and you weren't coming here to focus on hippos.
No, I'm here for work on rhinoceroses.
So tell us about that. What you plan on doing, what you're doing, and then we'll kind of get into how you ended up with hippos.
We're back to sperm again because I focus on rhinoceros sperm, and I'm part of a very large project, American Institute for Rhinoceros Science, or AIRS as we call it. And I'm part of the reproduction pillar. And my particular interest is I'm trying to figure out how do we sex sort sperm.
Oh.
So basically we want to pre-select the gender of the baby before, we inseminate the female.
That is
Interesting.
incredible.
I hadn't heard that.
Not me either. That is wild stuff.
Wow. Just because it's more important is it more important to have more females at this point?
Well, it
or
depends
just in
on the
future?
species, but
Okay.
for like rhinoceroses, they tend to be, females will group together, males tend to be solitary. So, and when you have a male, you tend to have a separate space for them and stuff like that. And also, I mean, we have a very small population, and we're trying to increase genetic diversity and stuff like that. And in those years where we have more males born than females, it can be kind of difficult because you kind of need females to help propagate. I mean, you need the males, but a little bit goes a long way kind of thing with male.
Yeah.
Females have to work a little bit harder.
Right.
I mean, especially since they gestate anywhere from 15 to 17 months long. I mean, that's a pretty long gestation, and plus you got to wait for the calf to grow up. And so the more females, sometimes it'd be better to make sure that genetic population stays stable and stuff like that.
That makes sense.
So, and what I'm hoping that if I figure out something for the rhinos, that it will be applicable to other species like elephants, who are also, a female-oriented group, that will do groups with females a little easier than they will in males and stuff like that. So
where do you even begin the process to sort
I don't
sperm
understand.
cells by male and female?
well, okay, so it's actually the sperm that decides if it's going to be a male or female. You've got the sex chromosomes. So there's sperm that carry the X, which is going to be a female, and there's sperm that carry the Y, and that's going to be the male. And right now, they actually have been doing it for decades in cattle because they use a fluorescent dye that marks that DNA, so something on the inside of the cell.
How do they find a dye that marks that specific DNA?
Like I said, us molecular biologists, we like to take things apart and make it glow. So we figured out how to dye DNA, and so it's doable. I mean, but it takes a lot of time, and it takes a lot of expertise, and it takes a very expensive piece of equipment.
Okay.
And we're talking hundreds of thousands of dollars.
Wow.
And like for cattle, they figured out, we only need like 4 million sperm. For when we talk about, rhinoceroses and elephants, we're probably going to need a couple billion sperm.
Wow.
So, while, we can sort a cattle semen sample in just a couple hours, it takes a little longer to sort. And while you have to look at each individual, we have an instrument that does it. There's not somebody with a scope going, you're an X, you're a Y. We've taught a computer and we've taught an instrument, but they still look at each sperm at a time. And a lot of sperm, and the other thing, this is the really frustrating part, if you're thinking about, endangered wildlife, about 3/4 of those sperm samples get rejected because they're like, I don't know.
Oh my goodness.
And with cows, we have plenty of cows. It's really easy to go, hey, I need another sample. Can you get that from the bull for me? With rhinoceroses, I'm kind of like every sperm is kind of precious.
Yes, definitely. Why do you need so many more for the rhinos or different species?
Well, we don't know.
Just for more testing or more data?
Well, no, it's just we know that when a male provides a sample, it has that many more sperm in it.
It's just a larger sample.
It's a very larger sample.
Okay.
And so you think about, in cattle, their reproductive tract is maybe, a foot and a half at the most. And like in an elephant, it's 10 feet.
Wow.
And it's,
so it's almost just size related.
Just size related.
Okay.
Cattle, we've worked for decades to figure out, hey, we can get away with a lot fewer sperm.
I see. Okay.
We're still working out, and we want to, let's try to mimic what happens in nature as much as possible.
Right.
So we want to have more sperm.
Okay.
And we don't want to be throwing anybody away.
No.
Yeah.
And we don't want to be damaging them. And that's the other thing. There's some slight level of damage. We kind of are hitting them on the head, and that's, they don't take that quite as well.
Yeah.
And like I said, we've had years to work with cattle and stuff. We're kind of, we can do it, rhinoceros. This amazing group at SeaWorld has figured it out, and they can do it.
Wow.
But my job is, can I figure out a way to do it that's not, we don't throw sperm away, that doesn't hurt them as much, and that anybody who can handle sperm can do it. Because right now it takes a very special expertise. So I'm trying to figure out if there's something on the outside of sperm. And based on cattle studies and horse studies and mouse sperm, there are differences between the male producing and the female on the outside of the sperm. Kind of the clothing they wear. You can think about some of them wear pink hats and some of them wear blue hats, kind of thing. So I'm trying to figure out if I can use magnets to go find me the pink hats. So I could just put the sperm in a tube and mix it with little magnetic beads that would attach to those little hats, and then put them on a magnet. So all those guys would stick to the side. I could pull out the ones wearing the blue hats, and then have a separated population.
That would be incredible.
That's my goal.
That would be
incredible. Now, whether I can make it happen, I don't know. I mean,
So
we're trying to do
that?
that in cattle and horses, and that's why I think it's feasible, because they are starting to do that with the domestic species. I'm trying also to do it with the rhinoceros.
And this would make it so much more accessible, right? Because you said right now you can do it, but you need these $100,000 instruments, and then
Wait.
if it's magnets, could anyone do it? Basically,
If you can handle sperm. And there's a little bit of training.
But magnets, how do you get them to just stick to the specific DNA? Where does that come from?
Well, it's so you're talking about proteins. Can be on the outside of cells. And then we have antibodies that would recognize those proteins. So you hook those antibodies to a magnetic bead, and then they would hook to the cell.
You can hook an antibody to a magnetic bead?
Oh yeah, we've been doing it for years.
Can you see it under a microscope?
Well, a powerful microscope. So
can you show me?
can you?
Well,
Yes, now. Can you do a video? What? This is incredible.
So,
can
I told you we were going to be learning a lot in this episode.
I haven't quite figured out the whole. Right now, I've figured out some proteins that look to be different between half the population of the sperm I'm looking at. So that suggests, because we know half of them should be X-bearing and half of them should be Y-bearing. Yeah. So if half of them are glowing because of my antibody and half of them are not, I'm like, So I'm actually going to go over to children's and use their cell sorter. So the kind of the same sorting, but I'm just going to separate based on the protein and say, okay, are all you guys my females and all you my males? Because if they aren't, then I will hook the magnets. But first, it takes a little bit of research to get to the simple part.
Yes, I believe that. No, that's not simple to me.
But that's what I do. I'm a geek. I'm a lab geek, and that's what I do.
I just don't understand. These are things that you can't even see with your eyes.
No,
Yeah.
A lot of the time I do stuff and I trust, and I think it's there, and until I make it glow, I don't know that it's there.
Oh my gosh! Wow, that's amazing. So you're working primarily on the Rhino Heirs project. And then we kind of drew you in. We've talked to Dr. Jessie Watuzik, who is no longer here at the Cincinnati Zoo. She, sort of accidentally also became a hippo person here. And so since she's not here, did you just, kind of... I don't mean roped into it. I'm sure you enjoyed the opportunity.
Well, I love the opportunity.
How did it fall on you?
Aaron asked, Dr. Curry asked me, hey, you want to come with me? We went on ultrasound Bibi. Because at that point, you guys were suspecting something was going on.
That was one of the very first ultrasounds, I remember.
Yeah, and because of my endocrine background, I could, I started thinking about, okay, so what's going on here? And that's when and I wanted to be part of the team of ultrasounding. It's something, I love being able to see what's going on the inside. So, and it just, was able to kind of start participating with that, and understanding. And I mean, I'm really interested in getting Bibi's spit so we can follow her hormones.
Yes, we've been collecting saliva from Bibi during pregnancy and now after also.
Yeah.
So you've been helping us with that.
Yes, because I mean, it's our way, hormones that, we tend to think for humans, we'll get a blood sample. Well, it's not always so simple from animals. And luckily for some of the species, we can get their spit or their poop, and we can measure their hormones. And their hormones will tell us if she's cycling or not. So, and during pregnancy, it tells us whether she's producing her own progesterone, which is that hormone that supports pregnancy. And it's also kind of the hormone we use to try to mimic pregnancy and keep them from getting pregnant. So
I'd like to talk about that a little bit because, so what happened when we first met Dr. Louisa, she came down to ultrasound Bibi to find out if she was pregnant. And it was one of those things where I was worried because I knew she was on birth control and I didn't know how that would affect the baby if there was one. But you made me feel better. So can you go ahead and kind of explain that?
Okay, so the birth control that we're using, like I said, it's basically mimicking progesterone. So it's trying to tell the body, hey, you're kind of pregnant, so you don't need to worry about cycling. You don't need to worry about making an ovocyte ready to be fertilized.
And then if they're not doing that, then they essentially
can't get pregnant. Then they can get pregnant. They're not listening to that hormone. They're like, I'm ignoring this phone call. I've got other things to do. If the brain has a different plan. So when you normally get pregnant, your body does produce progesterone. So like being on MGA, being on that progesterone mimic, it wasn't going to be harmful if she was pregnant because it actually could be supportive to it.
And that's actually something that is really possibly important to this pregnancy because looking back on the data or information we were able to collect from her first pregnancy with Fiona, it does sound like there was potentially low progesterone involved, and that could have been why Fiona was born prematurely, correct?
That we suspect that.
And we didn't know that until this second pregnancy. Like, none of us keepers realized that you guys had that information, or I don't know if it wasn't looked at until this second pregnancy. But people ask us all the time, like, why was Fiona born prematurely? And we're like, we have no idea.
I mean, it's a theory. I mean, we have one little piece of information. I mean, unfortunately, we don't know how often this may occur out in the wild if they, and stuff, or if this is a unique Bibi thing or something like that. But it's a theory because we know that progesterone is kind of supporting the growth of that baby. And like if her progesterone is low, that would explain why Fiona was so small. And that, for whatever reason, she gave birth early. And I mean, I don't know, can't really study that easily.
Right.
Like you said, there's not a lot that we...
all we know, it's just one in this instance. But
And so that's why, I mean, we were very encouraging to continue Bibi on something to help support that pregnancy so we didn't have that happen again.
Yeah, I was going to say you were able to use that kind of past data to make different management decisions with Bibi this pregnancy. Will you talk a little bit about how she was put on a supplement, the reasoning behind that and everything?
Yeah, so, I mean, because her progesterone level, and when Dr. Jessie was studying, she was able to get saliva from hippos from other zoos, and there was another hippo, from an early pregnancy whose progesterone levels were much higher than Bibi. So that's why we had an idea that Bibi's levels were lower than would be normal. And then, so the decision was made to put Bibi on another progesterone that has been shown in other species to be supportive of pregnancy, and that we knew wouldn't be a problem. So, because we didn't want to have that happen again. We don't know. I mean, our initial samples before we put her on suggested that she may be higher than she was with Fiona, but maybe it wasn't necessarily as high as that has been seen in other hippos. So we just really wanted to support that pregnancy. So we made the management decision, and it was a whole team, and I was not, I helped facilitate and advise. I did not make the decision entirely. I mean, lots of minds together, looking at other data and talking about what would be good for Bibi and this pregnancy. Put her on the progesterone, and they do it with women. They'll put them on progesterone to help support them and stuff like that. So, but we did, we didn't leave her on it the whole time. We did withdraw when we got near that, when we got past that window, when we knew, what safe for her to give birth. So past when Fiona was born.
We kept calling it the Fiona window the
whole
time.
Fiona window. That early
Because, and the reason for that is really important because progesterone will sustain a pregnancy, and if we would keep her on that too long, the baby could be too big, overdue, and cause danger in a different way. More danger for Bibi.
Maybe more for Bibi and the baby.
Well, yes,
if the baby gets too big, then she wouldn't be able to. It could be a very dangerous birth. I mean, that happens, in a lot of species. That if a pregnancy goes too long, then you have a too large of a baby and it needs assistance. And we really want to be as hands-off as possible. Yeah. So we withdrew her down. So we still haven't looked at the data, but you collected, her saliva the whole time. So we can look back to see what her progesterone levels were and kind of get an idea of how she did while she was on the supplement and then how she did when it was off. Because, she gave birth to Fritz basically almost within the 2 or 3 days I was predicting that she would.
Which is really cool, and something else that you found out because you started researching more, looking into it and talking to more people, and you taught me something that we've always said they have an 8-month gestation. But essentially, it's not super uncommon, or it's happened where they are born closer to 7 months and still, considered full term.
Still full
healthy,
term.
Not like the premature...
Not like Fiona was.
Fiona.
Right. So, it's just like with, we have a lot of data for human pregnancies. We say 36 to 39 weeks. And it's kind of the same thing with any species. And based on, I went into literature, what's been out there, and I kind of did some digging around and reading between all the sentences and stuff. And we said on average it's 8 months. But there's sometimes it can be, like you said, 210 days all the way to 240 days. So that's a month window of time. But the average is 233 days plus or minus 3. And if I remember right, Fritz was born, I think, on 230.
I wasn't counting the days, but he was born August 3rd. I don't know if that helps you.
But I think he was 230 or 230. He was there.
The 244 mark would have been August 15th because that was our original due date. So that makes sense.
So the 233, okay, maybe he was born 232 because I think I said August 4th is when I thought he was going to be born.
It was close. He was only about 2 hours late. Which, that's all very interesting and something that I also look at because then people, when we share this information, we haven't broadcasted it, not for any reason, we just haven't, but it's new information, it's interesting, but when people do hear it, they sometimes ask, so is Fiona not as premature as we thought? But I've always said she was anywhere from 6 to 10 weeks premature, possibly because Bibi and Henry bred for the entire month of July, and usually animals will stop breeding once an animal is pregnant.
Yes.
So the fact that Henry was still trying to breed Bibi at the end of July makes me think Fiona wasn't conceived at the beginning of July.
No, probably not.
And so that puts her at 10 weeks premature from the date we assumed. So anyways, she was still definitely premature and very small, and she couldn't
there was a
stand.
lot of things going on.
So there's definitely a difference with her no matter the dates or whatever, but it is interesting that you think you know a lot, and there's certain animals that I'm I know a lot of their natural history, and then there's still so much more to learn.
I had to learn a lot about hippos, but I enjoyed it. So I guess I think now I'm into this whole mega herbivores. I like elephants, I like rhinos, and I hippos.
Throw a little meerkat in there, and I'm good to go.
They seem kind of small on the small side compared to what I went from fruit flies all the way to the other.
Huge difference. So do you have any plans to continue other than the saliva we're collecting? Do you have time? I'm assuming the answer is no, but to start a project with hippos, or because the zoo, thanks to Bibi and her willingness to participate in a lot of things that hasn't always happened, which I think more zoos are doing way more with their hippos these days, just in general, and training with all animals and such. But Bibi's a good participant.
Yes. The way I approach this is I don't want to just do research for research sake. Okay. There's a lot of questions we can answer. Like I said, we've got a lot of black boxes when it comes to fertility and reproduction for a lot of our species. My point, I want to develop tools that help us support the animal's health that we need those answers to. So I kind of wait for you to tell me, I need to know this.
Okay.
And then I will help design the experiments and the research to help try to get those answers.
Okay.
So if there is something, and right now, we're trying to figure out what is the dose that we put Bibi on.
So can you tell me, is this dose working?
Right.
So we have changed the dose for Bibi, and it's probably more weight-based than we originally thought, or Bibi just needs a higher dose for some reason.
For whatever reason. And we may have to consider that she may need an alternative.
Oh, yeah.
That we're using. I mean, I've been talking to my colleague who is actually working on some of the birth control for those hippos down in Colombia.
Oh, very
wow.
Yes, I want to hear
The Escobar hippos, right?
Yes, the Escobar hippos.
Wow.
And so
You're
trying
talking
to
about male doing birth control for the males, right? Or some sort of
so
sterilization?
they're using, it's kind of a vaccine against one of the hormones that's in the brain that controls reproduction in both males and females. The gene RH. And so you can give it to males and females. You can give it to pregnant females, and it won't interfere with the pregnancy or anything like that. The idea, though, it keeps them keeps them from cycling.
Okay.
It's kind of like when you vaccinate against, COVID or something. You're like, okay, if this shows up, stop it. And that's like kind of that GnRH molecule is that key starting point to say, hey, I'm ready to produce either a female gamete or a male gamete, so we can have fertilization happen.
Okay.
So you can give it to both males and females. And I know there's not been a lot of work done on hippos, and it's kind of, they're trying to see, but...
Are we doing that with cats?
We're trying to do a non-surgical sterilant, but it's a different process.
Okay.
But it's the same kind of concept that you're knocking out that communication.
Okay.
And saying, okay, we're going to cut the phone line between you and say, okay, the gonads do not need to speak to the brain. We don't need any more communication. And so,
but that seems too easy, right? Like this vaccine. Why is it only working for specific animals, or why can it not work for?
So the thing with this is can kind of be a permanent thing.
Okay.
So it's like when you okay, we don't want you to breed ever again.
Okay.
And so that's why for a lot of the species that we work with that we manage, we don't really want to say no, you're done. When we're like, okay, we're just going to pause you because we need to maybe introduce you to somebody else or wait for your calf to be completely grown before we try.
Absolutely, but I'm thinking more of the stray cat dog overpopulation issues, and
that's why that's some of the research that's being done. Different scientists than me at Crew, but that's what they're working on because it'd be so much easier if we can just give a one injection, and then that's a lot less invasive, and then hopefully curb that population.
Right.
And it's not just here in the U.S. There's a lot of countries that struggle with an overpopulation of cats or dogs.
Right. Yeah, and I'm just thinking it should be in my mind easy. I don't know, to fix the problem. No suffering.
We have a goal. It's it's easy to think of. I just need a magnet and a little bit of organizer. But then I've got to find the connecting point, you know, and make sure it doesn't do anything else.
Right. Yeah. That's a good point.
So something I wanted to ask you, we had mentioned you were one of the main people that came over and helped with all these ultrasounds that we did. Jen and I have talked about what that kind of process was like for us, but you had a very different perspective while we were doing these ultrasounds. Will you tell us what that process was like for you on your side?
Well, I'm fairly new to ultrasound. We do it a lot in cattle and stuff, but I was so happy to join the team. And for us, we weren't sure what we were supposed to be seeing. And you look in we have guidelines, what we see in other species and stuff. And usually, like when cattle and horses, and which I've helped before, is you stick your arm, transrectally, because these are very large animals. They have got quite a large abdomen, so we can't do the same thing that we do with humans. But because of the unique nature of hippos and that very strong tail, that's not an option.
Yeah.
And Bibi's quite insulated. And so
That's a nice way to put it.
Well, she needs that insulation. She's going to be in water. Gotta keep warm. It's her own little wetsuit.
Right,
It's just several inches thick. And trying to bounce sound through that, it's kind of, it takes a team. And that's why we had a team that we come over with people, because we're having to press and and we need somebody to be watching the screen while somebody else is kind of underneath Bibi.
Yeah.
So, but she was incredible to work with.
So the person on the screen was kind of guiding you, left, right, up, down.
Well, it's not so much they would say, okay, that's a good spot, don't move, kind of thing. Give us a chance to look and see what, you're trying to watch over your shoulder, but a lot of times, you're under Bibi.
Yeah.
She's not a small girl.
No.
Yeah, so they're basically, I think we've talked about this before, but basically on their hands and knees on the floor, pressing as hard as they can against Bibi's, 2-inch thick skin, and then the layers of fat, and then trying to get through to her organs and find the right organ, and all of that.
With their arm straight out wide, as far as they can reach
like
the whole time.
I was like compared to holding like a milk jug, just straight for like 10 minutes or something. So, their arms get tired after a little bit, but then we're all like, but she's doing great. We want to see something. I'd be like, I just want to see the heartbeat. Like, we'd see if like somebody else could give it a try or whatever. But
Dr. Julie Barnes was she saw the heart, able to get the heartbeat, and she actually got.
That was my favorite video we've ever had. Yeah, so
that was awesome.
So, and it's, we have over at our Discovery Station. I don't know if you guys have come over and met Charlotte. So we have a black rhino that
I've heard about her, but we haven't.
You can ultrasound and kind of gives you the experience. Now, of course, that's transrectally and not abdominally, but it kind of gives you the idea because, small movements, you can see very different things as you're going through, and it's an art. There's science behind it, but there's an art to doing ultrasound. And so it's just, it was tiring, and we'd switch arms and switch people. That's why there's always a team of us working together.
I always felt for you guys, because I feel like I had the easiest job of the whole group. I'm just feeding her at the front, keeping Bibi happy, and you guys are sweating your butts off on your hands and knees, like, man.
Oh, man. No, after seeing those, it's so fun. I'm like, this could be my next career, a sonographer. I'm sure that's stressful too, but that's one of the best things these days is being able to do ultrasounds on animals. And knowing for sure that way, but of course you guys have developed ways to tell from saliva or fecal samples.
And we try to be optimistic about, we think that's a pregnancy, because for a lot of these species we're like, well, that's where the uterus is supposed to be. Right. That looks kind of like a uterus. Let's hope that's what, but for a lot of it's brand new information. It's just kind of something, and not a lot of people have done it yet. But like you said, more and more zoos are starting to do it and starting to incorporate it. Like, hey, let's monitor it. Let's see what we can see. So,
and then we mentioned we're still collecting saliva samples so we can see what Bibi's progesterone levels are like when she is not pregnant, and then we can use that in the future again. If she is pregnant again, we'll be able to compare it and see like the differences. And then will it also help us determine if this new dosage of birth control is correct for her?
Yeah. So the assay that we use measures her normal progesterone that she makes. Does not measure the supplement you're giving her. So we can tell, and the whole idea is that she will not be making any progesterone, because like when she's making progesterone, that means she's cycling.
Okay.
So, and we don't want her to be cycling, because that means she may be making a follicle.
So you're testing her saliva to see if there's any progesterone in it, and if there is, she is cycling and therefore could get pregnant.
Possibly, yeah. And there are some, I've been looking at the literature, then it does look like you can get small spikes in progesterone, but the cycle length is much shorter, so they're probably not as fertile if you've suppressed it somewhat. Yeah. And based on what we've seen, what little we've done with Fiona, because you'll be able to get poop from her, it does look like we've suppressed her progesterone, so she can't get pregnant.
Good. Which is good for Fiona. She's not ready for that.
No.
I'm not
Our little princess.
But truly, she's not big enough. We would worry about her size.
Yeah, she's a very small.
Yes.
Definitely. I feel like this whole kind of process has been a really cool collaboration, though, because we're able to kind of learn what we did from Fiona's pregnancy and, Bibi's pregnancy up with Fiona, and kind of make adjustments to this time around, and it worked like a charm, like you said. You had predicted Fritz's birth almost to the day, basically.
That's impressive.
And Bibi was amazing throughout the whole thing, carried a full-term healthy calf, and now we've got Fritz bouncing around the exhibit as proof of it.
Yeah. And every little... I know that we can be kind of annoying as we're like, well, can you get us more spit? Can you get us more poop? Can we come in ultrasound? But for us, it's like every little bit of information we build it. We build on what we don't know.
Definitely.
And then we can share it with others, so that if they find themselves, they're not like us. Like, has anyone else had this issue?
Right? Anyone?
So, and that's a lot what we do is like, so when someone says, something funny's going on, we say, okay. What questions can we ask? What tests can we do to try to figure out what's going on here, especially from the repro side?
Right. No, we don't mind at all. It's especially the saliva is so easy, and the ultrasounds are always fun. But the poop is different because hippos poop in the water most often. So collecting it and just depending on where Fiona sleeps at night and that sort of thing can make it difficult. But otherwise, it's very easy. Hippos poop a lot, so we don't mind at all.
And we always like to contribute to that stuff.
Yeah,
like we like to think that we're the samples and data that we're helping collect. Is
you
making a
would
difference
definitely
for the species, but,
Yes, for sure.
Definitely. So now that the hippo's all finished for the moment, you're back to rhinos.
Well, I'm always rhinos.
You're always rhinos.
And I mean, the hippos aren't done. We're gonna, like I said, we've got to keep an eye on Bibi and stuff like that. But I mean, most of my time is rhinos. But like I said, I do, depending on the needs that come up. When we have, people ask different questions and stuff, that we'll shift gears and we allot our time as needed and stuff. So it's it's a balancing act sometimes. My priority is rhinos, but when you have an issue, we come to the rescue. We help.
We appreciated that.
Or an unexpected pregnancy.
Yeah, exactly.
Surprise.
Was there anything else that we haven't touched on that you think would be really cool to share, or that you wanted to talk about with your job? I mean, magnetic.
I think you
antibodies.
We
and
went a lot of
proteins.
detail. I was going to say, you get me started talking, I may not shut up. I'm a science geek. I know I can get very sciencey and stuff, and I try to be careful.
No, you explained a lot of it really well. So, I mean, there's things that my brain just can't fathom, but that was great.
I feel like we covered a lot about me.
Yeah, definitely. All right, well then, should we embarrass ourselves with trivia?
I do have trivia for you guys.
Okay.
Obviously, we've got a reproductive specialist on the show today, so we've got some animal gestation slash incubation questions.
Okay.
So we talked about hippos, right at that 8-month window, but that's kind of an average, more like 7.5 to 8-ish months, somewhere in that range. So we're going to start. First question is an animal that's a similar size, a little bit smaller than a hippo, the black rhino. I imagine Louisa knows the answer to this one.
I know the answer to this. In fact, I may have mentioned it earlier.
I was going to say, I think I do too.
So what is the gestation period for a black rhino?
I was going to say 15 to 16 months, but I think you said 15 to 17.
Well, I was encompassing all the rhino species.
Oh, okay. So is black rhino 15 to 16?
15 to 16.
You guys are right on the money. 15 to 16 months. I should mention, all these species that I've got listed, by the way, are species that we've either recently had births or we're trying to reproduce and have births with. So black rhino, obviously we've got a couple calves in the past 5 or 6 years now with Kendi and a Johnny Joe.
Yeah.
But you guys are right on the money, 15 to 16 months. Calf is usually born at about 80 to 110 pounds. I think that's just fascinating because, like I said, they're a really similar size to hippos, but
double
twice the gestation period.
That's a good point. Yeah, I'm always blown away at hippos being so short. Do you have any ideas as to why that is?
Oh, no.
Okay.
I haven't done a lot of research into understanding why gestation is the way it is.
Yeah.
I mean, we know for, humans that, there's a lot, it has to do about brain size, the lengths of that, and then how much care they need afterwards kind of thing. Like how many hit the ground running and ready to go versus how much, and it may be, I wonder if it's more tied to hippos tend to be more in the water, and that's very supportive. You're not really that different from being in the uterus,
in the womb,
kind of thing.
Yeah.
Very supportive. Well, with a rhino, they're hitting dry land, and they have to be able to move it kind of thing, and get out of the way of things. So they maybe need to be more ready to go, and may develop that skin a little thicker. I don't know. That would be my hypothesis.
Okay, I like it.
That checks out with me. Next up, we got red panda. We've had many, many cubs here over the years. What is the gestation period for red panda?
I would say, isn't it about 2 months? About 60 days?
I was going to guess 3 months.
You guys are both a little bit low. It's closer to 4 months. They say it runs about 110 to 140 days, somewhere in that range. So just under 4 months. They usually give birth to 1 to 4 cubs, and the cubs are born at about 3 to 5 ounces. If you've ever seen them, they're just a little puffball. Yeah, they're these tiny little things with tons of fur.
Oh, that's so small.
Adorable, yeah. All right, next up, another one that we've had many, many babies over the years, little blue penguin chicks.
Oh.
What is the incubation period
for
a little blue penguin egg? We're jumping.
This is jumping species here.
Big time,
I was going to say, I can do mammals pretty well, I think, most of the time.
This is going to be a complete guess.
Oh, okay, I'm going to have to say, it's only like, 45 days, 60 days, maybe? I don't know.
45, 60
I don't know.
days, Jenna?
I was going to guess 67.
Luisa's right on the money. It's about 35 to
is
45
so little.
days. Yeah.
There's so not many
Yeah.
days.
Just a little bit over a month.
Well, and that you only have so much space in that little egg.
What's a chicken or a turkey?
I think chicken's about 20, 21 days,
Yeah.
something right there. Yeah,
Okay,
3,
that's
4
not
weeks,
long at
somewhere in there,
all.
yeah.
Birds do it quick.
Yeah.
Well, you think about who's going to come and raid your nests and stuff.
Right,
And so
yeah.
you've got to get that done really quick.
Yeah. The little blue penguin chick's usually born about 30 to 40 grams, about the size of a Hershey bar. For comparison.
Cute.
But recently correct me if I'm wrong, wasn't our last chick Rose after Betty White? I think that was our last little blue penguin chick.
I don't know.
I don't
I'll be really
know.
honest
Yeah,
with
she was
you. I have
born
no
over the
idea.
winter. I think she was named Rose, though, after Betty White. But moving on, next one we've got, it's been a few years since we've had a baby, but aardvark.
I don't know.
I feel like I should know this. I was going to say 6 months.
6 months?
Mm-hmm.
I think I'm more, let's see. I guess maybe about 5 months. I
5
think.
months?
Yeah.
You guys are both just a little bit low again. It's actually about 7 months.
Okay.
7 months.
Yeah, 210 to 220 days, somewhere in that range, which is crazy to me because
that's
right around
something
the
at the
hippo.
hippo.
Yeah. And an aardvark baby is born at about 4 to 5 pounds. So 7 months for a 4 to 5 pound baby. What was Fritz just born at? After 8 months, he was 80 pounds.
Yeah.
Exactly. Like that is
That's wild.
wild. All right, we've got one way off the beaten path here. An American burying beetle egg. How long does it take to hatch?
I'm sure Mandy taught us this.
I think Mandy
should
taught
know.
us this. Yeah, I think.
2 weeks?
Egg to hatch. So this is egg to larva
Egg to
stage?
larva, yeah, egg to larva.
Oh, then that's
Yeah.
that's really quick, isn't it?
Oh my gosh. I should know this.
I have no clue.
Egg to larva. I'm going to say like 24 hours.
24 hours, Lucy?
Well, I'll guess 36.
36. You guys, 36 is pretty close. It's about 3 days.
Okay.
Right at 3
72
days.
hours?
Yeah, about 72 hours. But that's a quick turnaround.
Yeah,
That's really
All right, last one up for you guys. Another species that we've had a ton of success here at the Cincinnati Zoo. The western lowland gorilla. We're back to mammals now.
Are they human-like?
There should be like human-like. I'm trying to remember. It isn't. It's about the 9 months, isn't it?
I was going to say 10.
It's closer to 9. It's about 8.5 to 9 months.
So
yeah.
it's less
Yeah, it's
than-
like the
okay.
human.
Just
yeah.
under human gestation. Yep. Yep, and their babies are usually born at about 3 to 5 pounds. I think our last baby was Elle here, right?
You're really asking me questions I don't... Was it Cup of Joe Burrow was the last little blue penguin?
Cup of Joe Burrow was the last blue penguin.
last
You're
blue
right. You're right. I forgot because that was right around the Super Bowl.
Yes.
Yes.
When,
yes,
Cup of Joe
yes,
hatched.
yes.
Yeah.
And
You're
then,
right.
yeah, Ella's the youngest, right?
Yeah.
Because there's Gladys and Mona, but they are older.
They're older than
Okay,
Elle, yeah.
yes, Ella's sweet.
Yeah. Honestly, though, I'm impressed. I threw a bunch of random species at you guys.
I got that
Lots of guessing happened. Okay, so we'll finish out the episode asking, what can I do? What is something someone can do that maybe you do at home, or
Okay.
you can share this?
What I'd like to share and encourage people to do is to compost. So I like taking those food scraps, like not meat or fat, but all the like when I make a salad, all those little things I chop off and don't use, and put it in a compost bin. Now I have my own little tumbling bin out back, and I use that to make soil to grow more things. But my point is that you can just kind of continue that cycle of life and not produce less waste. That we throw away less things that go into the landfill. We have a Keurig, and I love having my single cup of coffee, but I do try to make sure that I throw as little of it away as possible. So I've been trying to buy those pods that actually can be composted. And
very cool.
Yeah, I found a company that, like, even the bags the pods come in, I can throw in my composter.
That's
so
awesome.
like almost all of it breaks down except the little plastic ring, and they've told me it's because I'm not an industrial composter. I don't get hot enough. But I know there's companies here in Cincinnati that give you the opportunity if you wanted to be part of like an industrial compost and stuff, and that it may cost a little bit, but in fact it maybe save money and save the planet just a little bit at a time, because the less that we put in landfills, because that's doesn't really break down right. But like food in like a composter breaks down.
Do you understand it enough to explain that why putting this food in a composter and the heat and everything helps break it down, but when it's in a giant pile in a landfill, why it doesn't break down? That must get hot also,
well,
right?
It gets hot, but it's not just food. So you've got all these layers of plastics and metals and all the, and then, so like in a composter, we layer like the green stuff like from your kitchen, and we layer it with brown stuff like leaves or cardboard and stuff. And then we introduce like worms, because they're going to help move through it and break down. Then you have those nice bacteria and stuff. And a lot in the landfill, there's a lot of things that will kill all those things that would help break down the food. So you don't get we add air, we add moisture into the composter. So it's a whole ecosystem inside of itself to break it down and make dirt again and stuff. Landfill, we can't really do it, and they tend not to like stir it. Right? We stir our compost and stuff. So, and I mean, you just create these big piles that just they're stagnant.
Yeah.
They don't do anything. I mean, sometimes things break down, but not in the most pleasant way, and it's not just food things that make sense. So if we separate it out, we produce smaller piles of things, and everything, we recycle, all the plastic and try to recycle the metals and stuff. So what we put in the landfill is as small as possible.
I love that because, well, there's so much I need to learn about composting, and I think we could have one of our sustainability team members come in and tell us an entire episode worth. But it is interesting to know that you can put certain things and others, but there is a little bit of work that goes into it, but it's not too much work,
it's
right?
really not that
how
much
much time
work.
do you think you put into it in a week or whatever?
In a week, I mean, maybe 15 minutes. Because basically what I do is I collect it together until I get, like, I have a old kitty litter pail that I collect all my food stuff. And then once that's full, I take it out of my composter, and then I kind of put it in. I've got one of those ones that tumbles. So that, I kind of...
Does it for you instead of having to use a shovel.
Yeah, I just roll it down the hill and up again, and it's done for me, kind of thing. But there's a lot of different ways, and there's a lot of information out there. And like I said, I haven't looked at it too closely, but I think there's a couple companies where, like, they will give you a pail and you fill it up, they take it away, and then when you need compost, they bring you compost
very
back.
cool.
So, I mean, like I said, there's a charge with it, but I think it could be really worth it.
Yeah.
I mean, the plants I've grown with it, whether, it's just flowers or, just greeneries, or like when I grow tomatoes and stuff, they grow so much better and then I'm not going and buying fertilizer.
Right.
I've created my own dirt.
Exactly.
And it's really healthy dirt. Yeah,
it's really healthy
soil.
dirt.
Yeah, and that's why you get
and
the
the
better
worms
flowers
seem
and
really
tomatoes.
happy about it.
That's a great one that I don't think we've talked about before, and like I said, we'll have to do a whole episode on it, I feel like, but
that's another one of those things, like you said, every little bit counts, and every bucket that we can keep out of the landfill matters, right?
Right.
Yeah.
Every little action adds up. It makes, we're all greater, together than just, keeping it separate. You don't have to have, big actions are lovely if we can do it, but as an individual, it's usually the small things that are going to make the biggest difference.
Exactly. And that's one of those things I personally do not compost because it sounds intimidating to me, but I don't think obviously you just taught me and there's so much to learn, but I think with just a tiny bit of research and maybe an initial cost to get things you need. But like you said, use a kitty litter bucket and then the compost bin. So there's ways about it for sure.
Right. And there's a lot of options out there, and there's a lot of different ways depending on what space you have and what you do. And there's a lot of community gardens, and they may be able to work with them to may have a drop off to maybe it's a project we work to get buildings, so a neighborhood could just we all compost together, so then nobody has to have individual,
right?
And we spread the love around.
Yes, definitely.
Yeah.
Great. Well, I love it. I need to look into composting now.
I feel you. I'm right there with you.
I'm glad I could help.
Well, thank you so much for being here and teaching us about so many amazing things and helping out with the whole Bibi pregnancy. That was great.
I've had a
Fritz
lot of
is
fun.
doing really
Thank
well.
you.
Yes.
Yeah. It's been fun working with you over the past few months with the pregnancy, obviously. It's been fun having you on the show. We know you're busy. We really appreciate you taking the time to join us.
I've really enjoyed this. Thank you.
Yeah.
All right,
so
everyone.
much.
Thanks for listening, and we'll talk to you next time.
Yep. Take care, guys.